Alright, so we need to understand what a cell is, the types of cells, and also the structures which are within these different types of cells. Now, a cell is defined as a basic structural and functional biological unit of living organisms. The basic structural, functional, and biological unit of living organisms. So they are often called as or referred to as basic building blocks of life. So the cells, we need to know that they consist of the protoplasm, which is the living part of the cell, and the protoplasm is surrounded by a membrane. Okay. Now, this living part of the cell called the protoplasm is made up of the cell membrane, is made up of the nucleus, and also the cytoplasm, those three parts. So we need to take note of them: cell membrane, the cytoplasm, and the nucleus. So living organisms are either unicellular or multicellular. So just from the word uni, what do we think it's talking about? What does unicellular mean? So we're talking of cellular, but uni. So when we're talking about universe, uni, so it has to do with one. So unicellular means most living organisms are unicellular, meaning they are single cell, they only have one cell. That's the meaning of unicellular. And then others are multicellular, meaning they've got more than one cell. So examples of single-celled organisms are either unicellular or multicellular, not most of them being unicellular. Actually, most of them are multicellular. Now, an example of an organism that has got only a single cell, like the whole body is only one single cell, is amoeba. And then multicellular cells or multicellular organisms, these include plants, animals, and most of the things that we know, those are multicellular. All right. So now again on the types of cells, cells can either be prokaryotic or eukaryotic. That is also another classification. Well, when you are talking about prokaryotic cells are the oldest cells. Actually, these cells existed before eukaryotic cells, and eukaryotic cells are believed to have come from prokaryotic cells. So prokaryotic cells, pro means before. Karyot is more like the nucleus. So prokaryotic means before the nucleus. And then eukaryotic cells, these are cells which developed and the nucleus was known. So prokaryotic cells, one characteristic of them is that they do not have a nucleus and they do not have membrane bound organelles. Their organelles are not bound in membranes. Now, when you're talking about eukaryotic cells have got a nucleus, a true nucleus, and their organelles are bound by membranes. That is another thing that we need to know. When it comes to questions where they're going to ask you to describe, you need to understand this because they ask for a description and you don't know how to describe. How do you just get to go about it? So you need to know this. Prokaryotic cells are the earliest known cells. Okay. So when you're studying biology, we believe that cells were discovered by Robert Hooke. So that's the person who discovered these cells that we're talking about, Robert Hooke. Alright, so now we need to understand the prokaryotic cells and the eukaryotic cells in details. So we're going to start with understanding of a prokaryotic cell. How is the structure? what are the parts in a prokaryotic cell and what are the functions of a prokaryotic cell. So I'm going to put down a structure that is going to help us be able to tell how a prokaryotic cell is. Firstly, a prokaryotic cell has got a cell membrane, that is the outermost shell. It has more a definite shape like an animal cell. Okay. And then it has got also another inner membrane there. Now, just before having the cell membrane, the outermost part of a prokaryotic cell has got is covered in what is called uh, a cuticle. I mean a capsule. That's the outermost shell. You can think of also like a leaf. There is that shine surface, the outer part of a leaf that is shiny. It's more like grayish like that. That's the outermost shell, or outermost part also of a prokaryotic cell. And then now we have got a cell wall. This thick thing that we're able to see, that's the cell wall. Well, for prokaryotic cells, they've got more like hair structures which enable them to move. So we have got a much longer hair structure which enables a prokaryotic cell to move. It can be one or there can be a lot of them, and that's called flagellum. There. So it enables the cells to be able to move, these prokaryotic cells. And then we have got even more other hair-like structures, but they are much shorter and they are much numerous in them. Like there are a lot of them. And these others, yes, Paxina, go on.
These prokaryotic cells actually found in amino, I don't know amino whatsoever.
Amoeba.
Do they have do they have cells? This cell is only found in a plant cell.
Okay, good. That's a nice question. So when you're talking about the types of cells, we have got two types of cells: the prokaryotic cells and the eukaryotic cells. When you are talking about the classification of cells according to, I mean, the classification of organisms according to the number of cells that they have, you can now classify them as either unicellular or multicellular. So it is in the unicellular that we have amoeba, meaning it has only a single cell. The whole organism has got only one cell. So prokaryotic cells are not examples of amoeba, let me say in that case. Does that make sense?
Yes.
Okay. So these short structures which are able to see air-like structures which are also used for more like feeding and also for movement, these we call them as pili. Okay. Now we are going to go inside this cell. Inside we are going to have the cell membrane, which allows what enters inside and out leaves the cell. So that's the cell membrane. And then inside, every organism should have DNA. But if we talk about humans and plants, the DNA is stored in the nucleus. Now, because prokaryotic cells do not have a nucleus, their DNA is just within the cytoplasm. So, this region we can see here, that's the cytoplasm. So, their DNA is just in extended in the cytoplasm. we're going to understand more about DNA, its composition later. It's a topic on its own. And so this, when the DNA is folds and is together, that is what we call now a nucleoid. So there is no nucleus, but there is a nucleoid, just a region where DNA is found just within the cytoplasm of this cell. Apart from that, we also have ribosomes just within the cell. So ribosomes are there for protein synthesis when we want to form proteins. So these are just small parts which we are able to see inside there. So apart from that, we also have, of course, just understand we're talking about parts. So there can be different parts of this prokaryotic cell, but these we've talked about here are basically the most important parts. And another part that you need to know is we also have another DNA that is free, that is not part of the DNA that is found in the nucleoid, and that is called the plasmid. So the plasmid also contains DNA. We have got what is called plasmid DNA. So DNA is what affects more like everything about a person. It will determine, or it will talk about why do you look like your parents. Why is it that some sicknesses are carried on from uh, parents to offsprings? Why is it that there are some traits which are just within a family? So all those things are carried in the DNA. Now the plasmid DNA does not have more effect on the organism, but the plasmids also contain DNA. Alright. Now, just as cytoplasm here contains what we call cytosol. So cytosol is more a fluid like uh, portion in the cytoplasm, the fluid like part. That's what we call the cytosol. That is where all these organelles we are saying the ribosomes and these other things are dissolved. All right. Now let's look at this in details a bit, starting with the cell wall. So the cell wall gives rigidity and shape to the bacterium. This shape you can see here is maintained by the cell. The rigidity is because of the cell that is present. That is why you can even see plants, they can be able to stand up straight because of the cell wall. So the importance of the cell is that it prevents the cell from being able to swell and burst. So even if there is an excessive buildup of water in the cell is not going to be able to burst. Why? Because of the cell wall. Okay. That is in the process of course osmosis. Osmosis, the movement of water inside and outside of the cell. So another thing that we need to be able to know is that when you're talking about just the cell wall, the cell has a substance which we call as mirin. So mirin is more a polysaccharide. Polysaccharide meaning we just have more monosaccharides or sugars together. So it consists of polysaccharides which are cross-linked by short peptide chains. When you are talking about peptide, this is just meaning protein. So bonds which are found in proteins are called peptide bonds. So when proteins are being made, first we are going to have peptides. Now, when these peptides are modified, then we are going to call them as proteins. They are modified. Modification has to do, a protein has to fold itself into a certain shape. After the folding, then it's going to be known as a protein. So mirin is made up of polysaccharides cross-linked with more like proteins. Now we are calling them peptides. All right. This is what makes up what? Bacterial cell walls. You need to know because the cell of a bacteria is different from a cell of a eukaryotic cell. Well, I'm talking about bacteria because one example of a prokaryotic cell is a bacteria. That is why I'm referring to bacteria instead of mentioning prokaryotic cell. So they are made up of murin. Now, just the cell wall is going to determine the type of bacteria. We have got what we call gram positive bacteria and also gram negative bacteria. So an example of a gram negative bacteria that we talk about is the most common. This structure you can see here is the structure for E. coli bacteria. So because the name is a bit long, we don't finish the name, we just say E. coli, but it has got a name. The full name is, even the pronunciation of course is a little bit tricky. Let me just write it, perhaps you can read it better than I can do. So this E you can see, that's what it represents. Escherichia coli. Now, because the name is long, we'll just say E. coli. So that is one example of a bacteria, and that is gram negative. And then we have got gram positive bacteria, for example, lactobacillus. Now, what's the difference between the gram positive bacteria and the gram negative bacteria? So the difference is that when we are talking about the gram negative bacteria is that they cannot be stained by a gram stem. Cannot be stained by what? By what we call a gram stain. They cannot be stained. When you're talking about gram positive bacteria, being stained is more like, for example, if someone gets ink they put on you, the ink is able to remain. So gram negative bacteria, when you get this what we call gram stain, they will not have that color for the gram stem. But for the gram positive, they are able to be stained by the gram stain. So that's the difference. So all this, for you to be able to determine whether a bacteria is gram negative or gram positive, you are going to look at the cell of that bacteria. So if the cell can be stained, then it is called gram positive. If it cannot, then that is gram negative. Of course, the cell wall just allows the penetration of gram stain into the cell of this bacteria. So that is why it can either be stained or not stained. Alright. Another thing, we're talking about the cell membrane, which is also called the plasma membrane. Now, for this one, you need to understand more the structure because just the cell itself, the structure is a bit complex also. So the cell wall, I mean the cell membrane is made up of what is called a lipid bilayer. And when you're talking about bilayer, meaning we have got two layers. So what you are going to see is we've got lipids, more like that, on top or inside and outside. Now, this kind of lipids, they've got the head and the tail. And the tails, they point towards each other, while the heads are away from each other. We are going to look at this kind of lipid. So this is called a phospholipid. And phospholipid has got two parts. It is made up of a lipid and a phosphate group. That's why it is named as a phospholipid. Okay. Now, what is going to happen is this. The phosphate, which is the head in this case, so what you are seeing as the head here is called the phosphate group. The phosphate, which is the head, is charged. It can interact with water like a phosphate group is more like that with a negative charge. So it can interact with water because it has got a charge. But the tails, because they are lipids, remember lipids are hydrophobic, meaning they hate water. If you mix cooking oil with water, no matter how you mix them, they shall never mix. They hate water. So, because these parts hate water, that's why they are inside. They are staying away from the water. They stay away from the water. And then here, these parts which love water, they are exposed to the water. So, this part is outside the cell. And then this part is inside the cell. So inside the cell there is water. Even outside the cell there can be water. That's why the heads are outside and inside. But this part shouldn't interact with water. Okay. And then there are some other compounds which are just within the lipid bilayer. We have got glycolipids. Apart from glycolipids, we've got also glycoproteins just in the cell membrane there. Apart from that, there is also uh, cholesterol. Let me put this here as cholesterol. And we have got just other transport proteins. This big thing you can see here, let me put it like this. This is going to represent a transport protein, or let me say integral protein. We have got also proteins which are at the end of the cell membrane. These are called peripheral proteins at the end. Remember, this is just a cell membrane. So the cell membrane has got a lot of components. Now we need to know the function of all of these parts which we are seeing here. What's the function of the glycoproteins, glycolipids, cholesterol, and these other proteins that are found on the cell membrane? Okay. So the first thing is we are talking about phospholipid. We are saying it is made up of a phospholipid bilayer. So phospholipids provide barrier in cellular membranes to protect the cell, and they make barriers for the organelles within these cells. So these phospholipids which we can see here, they make sure that they provide a barrier so that things will not just be able to enter in and out of the cell. That's why we say the cell membrane is there for making sure that not everything that comes is going to enter or leave the cell. So the phospholipid is there. Now, when you are talking about charged and uncharged particles, well, uncharged particles can be able to pass through here, and charged particles can also be able to pass through. Now, remember, these are lipids. Because they are lipids, meaning they are hydrophobic, they hate water. So, just for water to be able to pass here, it's not that easy. So, because of the small shape for water, the small structure for water, that's why water is able to pass through. Otherwise, water can is very difficult to pass through because this portion is hydrophobic, it hates water. So in other words, since this is a lipid, lipids attract other lipids. So charged particles or charged molecules are very difficult to pass through the cell membrane. That is because it is uncharged. But uncharged molecules are able to pass through freely here. So for those molecules which are charged, they need some form of transportation because they are passing through uncharged medium. That's why we need transport proteins. That is one function of transport proteins which are found on the cell membrane, to transport ions and other molecules that are not able to pass because they are charged. Alright. So phospholipids work to provide pathway for various substances across the membrane. Phospholipids help cell membrane and membrane surrounding organelles to be flexible and not stiff. So it is the phospholipids which make these to be more flexible. That's why more like rubber, you can be able to turn it. They are not that stiff. Okay. So that is one function of these phospholipids we are talking about. Of course, other parts we are going to be able to talk about them, and you can be able to make even deeper and deeper researches. Now, we also have glycolipids here. Glycolipids are found on the extracellular faces. They are found at the more like at the ends of the cell membrane, and they are more found also in eukaryotic cells. Now, their function is to make sure that they maintain stability of the cell membrane and facilitate cell to cell recognition or interaction. So these glycolipids we are able to see here, they make sure that cells are able to recognize themselves and also to just maintain the stability of the cell. They can also act as receptors for viruses. So when viruses are about to come and maybe enter the cell, these are able to tell that they are viruses which have come. So they are there for reception. They are receptor parts also of the cell. So apart from viruses, they're able to also act as receptors for pathogens which enter the cell. Pathogens, these are just disease causing organisms which might be able to enter the cell. So they're able to track them out. So glycolipids are simply lipids with carbohydrates, just like we can hear from the name glyco, meaning coming from the uh, from carbohydrate, or let's say sugar, and then lipid. So we've said they facilitate cell recognition and cell interaction, which is very crucial in the way the cells are going to be able to respond, like in the immune system of the cell. If you are not able to recognize what is not part of you, even the immune system will not be able to work because they feel or they take it that everything is normal. So that is the importance. Okay. And they also allow for cells to be able to connect to one another and to form tissues. We also have glycoproteins here. Glycoproteins are also found on the surface of the lipid bilayer, as we can see just at the ends there. Now, their hydrophilic nature. So we are saying these are hydrophilic. Proteins, we need to know that proteins have got both hydrophobic and hydrophilic nature, meaning they can both hate water and interact with water at the same time. There are some portions which hate water, other portions love water. But generally, proteins love water. That's why they can also dissolve. So, because they are hydrophilic, meaning they love water, they allow an aqueous environment, and they act in cell to cell recognition and binding of other molecules. Because of the hydrophilic nature, that's why also some of them, some other proteins are used also for the passage of what? For the passage of those charged particles or charged ions. Glycoproteins are special proteins that have oligosaccharides. When you talk of oligosaccharides, these are just a lot of monosaccharides, about 3 to 10 monosaccharides. Those are what we call oligosaccharides. So we have got monosaccharides, disaccharides, oligosaccharides, and then polysaccharides. So these glycoproteins They form hydrogen bonds with water molecules surrounding the cell, and so they help to stabilize the structure of the cell membrane. So that is basically on glycoproteins. Now, another important thing that we have in the cell membrane is cholesterol. Cholesterol is so important, it plays an important role in the fluidity of the cell. So how it does this is when it is too hot, cholesterol is an example of a lipid. So what happens is when it is too hot, instead of the cell being able to melt, the cholesterol is going to come in and make sure that the cell maintains the rigidity. And when it is too cold, instead of the cell being able to more like freeze, cholesterol is going to come in and make sure that the cell doesn't freeze. So that is the importance of cholesterol. It is a very important molecule. So it is there for stability of the cell. All right. So these are some parts, and we also have different proteins like integral proteins, peripheral proteins. So membrane proteins, or these proteins which are found in the membrane, are very important for transporting of substances across the cell membrane. They can also act as enzymes or receptors on the extracellular fluid that is at the ends. Uh, they are there for cell recognition also when they get to combine like with carbohydrates forming glycoproteins, which we've already talked about that. So these are functions of proteins, and these are some parts which are found on the cell membrane. Apart from the cell membrane, we talked about the pili as one part of the prokaryotic cell. The pili, these are structures which are just found on the surface of the bacterial cell. They help different bacterial cells to stick to each other for the purpose of exchange of genetic materials during reproduction. So that is pili. Those small structures which I drew around the, which I lot around the cell. They can be sometimes used for movement in some prokaryotic cells. We also have the flagellum, which one that long structure which I showed, which is air-like. So this is a complex sweep-like structure made up of protein called flagellum. It's made up of a protein. The flagellum is made up of a protein which we call flagellin. So it helps bacteria to move around by the use of a motor protein that spins. So there is more like a motor protein that is going to be spinning. Because of that, then the bacteria is able to move. So the base of the flagellum rotates on ring-shaped bearing so that the flagellum performs rotation motion to push the cell forward. So they are more like a rotation that happens. There is more like a propeller there that makes the bacteria to be able to move just on the base of the flagellum. We also have the cytosol, the cytoplasm, which is the fluid content in the bacterial cell, enclosed by the cell membrane, the fluid which is inside the cell membrane. So it is the cytoplasm which contains ribosomes, DNA, enzymes, lipids, and also some droplets of carbohydrates. And these carbohydrates are also important in that they participate in cell recognition, and they are also there for structural roles and also as a physical barrier. Apart from that, we talked of the nucleoid, which was just the region where DNA is found. So it is just the nucleus is just a region in the cytoplasm which contains cellular double-stranded DNA. Now we need to know DNA is double-stranded, and we're going to talk about that. Okay, the DNA stores, of course, genetic information or genetic material that controls the cell, and this genetic material is going to be passed from the parents to the offsprings. So that is one thing. And then we also had plasmids. We said those plasmids also contain DNA. So plasmids are small pieces of DNA which are separate from the actual DNA, or what we call as chromosomal DNA. They usually replicate at the same time, so they also make a copy of themselves when the other DNA, which we call as the chromosomal DNA, is making a copy of themselves. So apart from that, we also have ribosomes, and we said ribosomes are used for protein synthesis. Now ribosomes, we have got the small ribosomes and the large ribosomes. We call them subunits. So depending with the kind of cell that you're talking about, in this case we're talking about prokaryotic cells. Let me just put them in advance so that you'll be able to remember. We've got prokaryotic cells and eukaryotic cells. So we have got the small subunits and also the large subunits. So for the prokaryotic cells, we have got the 30S and the 50S. So when these two combine, then we are going to form the whole ribosome, which is found in the prokaryotic cell, which is called the 70S. Now these are not mathematical calculations. All you need to know is that the small ribosomal subunits are called the 30S, the large ones are called the 50S. And then in eukaryotic cells, the small ones we call them as the 40S, the large ones they are called the 60S, and then when we combine these, they give us the 80S. One way you can know that is the small ones we call them 30. Just add the 20, that will give you 50, and then add the 20 to the 50, that will combine give you the combined form, which is the 70S. And then for the eukaryotic cells, the small ones are just 10 units more than the 30S, giving you the 40S. And then the 40S add the 20, that will give you a 60S. 60S add the 20, that will give you the 80S, which is combined now. So the S represents seed bag unit or seed bag constant, which just represents how much can this ribosome, yeah, the same small ribosomal subunits and the large ribosomal subunits, be able to sediment. When you are performing centrifugation, you want to rotate this or you want to spin it. How much are these ribosomes able to sediment? Go at the end. That is the seed bag unit. So these are some parts of the cell, and those are some functions.